Welcome to the National Invasive Species Awareness Week webinar series, hosted by the Southern IPM Center and sponsored by the University of Florida, Texas AgriLife Extension, Auburn University, and University of Georgia Center for Invasive Species and Ecosystem Health. Today we are proud to present Charudattan, talking about a novel bioherbicide for Solanum Viarum, tropical soda apple.
Good afternoon. Let me begin with today's invasive species. The topic of today's my talk, Solanum Viarum, or commonly known as tropical soda apple. It is a plant native to South America, mainly in Argentina, Brazil, Paraguay, and Uruguay, although it occurs now in many other South American countries. The plant was first reported in Florida in 1988 in South Florida. It now infests about 404,000 hectares in Florida alone. It's found chiefly in pastures and wooded areas on ranches and former cattle ranches that have been turned into conservation lands. Annual cost to landowners from this weed in control costs as well as lost revenue is estimated at about $6 to $16 million annually. It occurs in several southeastern U.S. states. A map from University of Georgia Bugwood site is perhaps the most current and accurate. It shows weed is present in Florida, Alabama and Mississippi, Louisiana to the west, Georgia, North and South Carolina, and Tennessee to the east and north, and Puerto Rico. There are a number of other maps that you will find on the website. Many of them are outdated. We certainly need the distribution records updated. According to CABI, the UK organization, the weed also occurs in 20 other countries around the world. It is an invasive or problematic weed in several other countries, including in Brazil, in improved pastures, and in Brazil the plant is native. The plant is cultivated in India for medicinal purposes as a medicinal plant, and there has been considerable work done on plant improvement in India. As far as I can tell, so far the plant is not reported as a weed in India. For us in the United States, tropical soda apple is a noxious weed, and in Australia, in New South Wales, it's classified as a Class 2 regionally prohibited weed. Here are some aspects of the plant's biology. It's a thorny plant, as you can see in the picture panels in this slide. The leaves and the entire shoot is covered with very sharp, painful thorns. Depending on the climatic zone and the seasonal conditions, the plant is either an annual or perennial. And although morphologically it is a herb, it can also assume a shrubby growth habit. The plant is toxic, as are many other members of Solanaceae. It is listed as a plant toxic to livestock. Certainly the foliage is unpalatable to cattle, but the cattle and possibly other grazing animals feed on the ripe, mature yellow fruits. The principal mode of dispersal is seed, mainly through cattle and other grazing animals that consume the fruit and spread the seeds. And also contaminated hay from tropical soda apple infested areas is also a major contributor to the distribution. The plant is a prolific seed producer. Up to 50,000 seeds per plant has been counted. For the current best management options, please see the University of Florida Center for Aquatic and Invasive Plant website. This is the most thoroughly tested comprehensive plan. Among the options we have are chemical herbicides, chiefly those containing aminopyrrolide, products such as Milestone, Grazon Next, Chaparral, all of these are Dow Agro products. Mowing where possible, and the mowing has to be repeated up to 3 times per season in most areas. And a classical biological control agent, Gracchiana boliviana, a chrysomeliid beetle. And the topic of today's talk, a new bioherbicide, SolviNix liquid concentrate. So what is SolviNix LC? It's a biological herbicide containing the liquid concentrate of a plant virus. The virus is tobacco mild green mosaic tobamovirus, in particular strain U2. It is a well-known, well-characterized virus found worldwide in Nicotiana tabacum, the cultivated tobacco, and Nicotiana glauca, tree tobacco. A naturally occurring isolate is used in the SolviNix LC. It is not genetically engineered in any way. SolviNix is labeled only for the control of Solanum Viarum, tropical sword apple. It is registered for use in pastures and wooded areas only that are fenced to contain grazing animals. It is registered under FIFRA Section 3, and it is registered in the state of Florida, and we are working to get it registered in other southern states where we are likely to use it. So what is the novelty of SolviNix LC? First of all, it is the world's first registered bioherbicide containing a plant virus as the active ingredient. The virus has a unique mode of herbicidal action. The virus elicits a hypersensitive resistance response. The hypersensitive response is typically a resistance response. But in TSA, or tropical sword apple, the resistance response, after an initial period of about a week or so, slowly starts to turn into a systemic necrotic necrosis reaction. Plant dies as a result. Unlike a chemical herbicide, which is typically sprayed to cover the entire plant canopy to deliver a desired active ingredient dose, SolviNix is applied to just a few leaves per plant, and from there the virus, the active ingredient, amplifies in planta and kills the plant. The HRSN, the hypersensitive response systemic necrosis interaction in TSA, is a genetically controlled plant virus interaction. It may be possible to exploit this interaction, genetic interaction, as an herbicidal mechanism of action. How does SolviNix LC work? The virus elicits the HR, which is visible as foliar local lesions or necrotic flecks, as you can see on the picture on the right. This happens about a week after inoculation. About 2 to 4 weeks later, 4 weeks after inoculation, the infected plant starts to wilt. And about 4 to 6 weeks after inoculation, the wilted plant starts to die. These reactions go like clockwork. They are predictable, consistent. As long as the infection has taken place, one is sure to find these reactions go according to what we are projecting here. TSA plants of all ages, from seedling stage to full maturity, are susceptible and are all killed. All parts of plants, including root system, is killed except the mature fruits. So far, we have not found any variability in the systemic necrosis response to the virus among TSA plants from the United States or a small group of accessions from Australia. So this species perhaps has no natural way around the systemic necrosis response. Here are some examples of TSA control with SolviNix LC applied with a backpack sprayer in pastures and biodiversity-rich conservation site. As you can see in the picture on the right on the top panel, plants treated with SolviNix compared to control plants. And the plants in the lower panel on the left were sprayed. The plants on the left, the tropical sought after plants, showed up in a freshly prepared ground for planting. And after 7 weeks, we had pretty much killed all the tropical sought after plants in this place, and grass had regrown and filled the field. On the right, a plant at the foot of cypress tree in a biodiversity-rich conservation site was sprayed, and 33 days later, the plant is dead, as you can see in the picture on the lower picture. There are two application tools that are highly effective. First of all, in order for SolviNix to be effective, the virus must penetrate the foliage. This is accomplished by abrading the foliage before spraying, or spraying at high pressure, about 80 psi at the point of impact, which forces the virus into the foliage. It literally makes the virus infiltrate the leaves. We have tried many other effective methods, particularly we are impressed with the wet blade mower as a means of application. But so far, EPA has allowed us to list only backpack sprayers and handheld herbicide wipers as approved tools for application. The SolviNix should be applied to actively growing plants. Plants that are dormant, either from cold temperature, drought, or flooding, will not react to the virus. The host-virus interaction will not proceed normally, and therefore the plant may not be killed. Ideally, the SolviNix should be applied before fruit set. Here are examples of the tools we have used. The backpack sprayers on the left are things that we put together. These are all material available off the shelf. The CO2 compressed air or CO2 cylinder can generate up to 200 psi, and so can the electric pump. It can also generate 200 psi. The handheld wiper on the right is produced by a company called Micron. It's called MicroVipe. These are effective. Now, SolviNix in its place in TSA management plans. TSA is difficult to control in wooded areas, under trees, and around wood piles and structures, as well as in biodiversity-rich conservation lands where chemical herbicides and mowing are difficult to apply. It may be undesirable or unacceptable to use chemical herbicides, which can leave residues in manure, milk, and meat. Chemical herbicides are not selective in pastures intercropped with clovers, perennial peanut, and other broadleaf forages. The beetle, which is good, which reduces plant fitness by affecting plant growth and number of fruits per plant, but it does not stop fruit set or seed buildup. The beetle certainly does not kill the plant. The beetle is generally less effective in regions north of latitude 29 north. So Here are examples of the way tropical soda apple is present these days. These are the pictures show the areas where the weed is kind of hard to control with some of the chemical and other methods. And the picture on the extreme right shows a mature plant fully loaded with Gratiana boliviana, but you can see the plant is still producing significant number of fruits. For the above reasons, SolviNix is a suitable additional or alternative option to manage TSA in southeastern United States. It can be used selectively to kill TSA without harming other desirable plants. It can be used in pastures intercrop with clovers, perennial peanut, and other forages. Being exempt from a tolerance requirement, it can be used without removing or holding cattle from treated areas. Residue in manure, meat, and milk is not an issue, and it is very unlikely that virus will show up. And by applying before seed set, it can be used to reduce or even stop further seed buildup in the field. But here are some key aspects to remember. SolviNix is composed of a mechanically transmitted virus. It is not transmitted by insects or other vectors and cannot self-disseminate. This is an important safety feature. SolviNix has no residual soil activity, and it is not labeled for soil application, so it won't do any good to apply to soil. It will not prevent seed germination or seedling establishment. For these reasons, it will be necessary to reapply SolviNix to control TSA plants that germinate and establish after a prior application. It may also be necessary to reapply to control plants that were missed in the prior application. But when properly applied, SolviNix can provide nearly 100% control of treated plants, that is 100% kill, thereby minimizing the need for reapplications. TSA fruit present when SolviNix is applied may or may not be killed, depending on their level of maturity. Mature fruits may ripen and produce viable seeds, even as the plant begins to die. Immature fruits may not die before the seeds mature. Therefore, for best results, SolviNix should be applied before the plant sets fruit. And this will prevent, hopefully, further addition to the soil seed bank. Is the plant host range of the virus TMGMV U2 a concern? TMGMV is adapted to plants in Solanaceae. In nature, it's found only in a few species in this family. However, like many other plant viruses, it can be artificially inoculated onto plants in several unrelated families. So some of the published host range lists that you find in the web for this virus, really, they come from artificial inoculations. The real natural spread of the virus is limited to just a few plants. Besides TSA, this virus can kill some but not all cultivars of capsicum species, that is annuum, frutescens, chinans, and so on, as well as Nicotiana species, tabacum, as well as some of the other Nicotianas, and Physalis pubescens. These are the only plants where we have seen actual plant kill. Our analysis of the potential risk of exposure of susceptible species to this virus did not reveal any undue risks to non-target plants. Most of the susceptible plants that we found, and those are listed in the some of the websites, they are unlikely to be present at sites where tropical soda apple is a problem. The little table at the bottom shows the summary of our host range data based on our tests with 435 species cultivar combinations. About 53% or 58% of the plants we tested are immune, 11% are resistant, about 9% are inconclusive, asymptomatic, and 15% are susceptible with non-lethal mosaic symptoms, and about 8% of the plants belonging to Capsicum, Nicotiana, Physalis, and Solanum Viarum die from this virus. Can this virus spread widely and uncontrollably as a result of its use as a bioherbicide? Again, the virus is a mechanically transmitted virus. It's not known to be transmitted by vectors. It's not seed-borne, and it's not pollen transmitted. Because of this, it's unlikely to spread linearly or exponentially in nature from the treated plants. And it will not spread in the field linearly or exponentially like, say, a whitefly transmitted virus can, because the whitefly acts as the agent of spread. The virus is prevalent in states where SolviNix is likely to be used. Unlike an exotic classical biocontrol agent, using this virus does not constitute releasing a new agent into the environment. It's already present here. For these reasons, as well as based on epidemiological theory and practical evidence, this virus cannot spread widely and uncontrollably as a result of its use as a bioherbicide. For further information, please visit my website, www.bioproducts.com, where you'll find more information about SolviNix, how it works, labels, safety data sheet, product brochure, etc. Thank you for your attention.
I'd like to thank you all for attending the webinar as part of National Invasive Species Awareness Week. The recordings of this webinar and others will be available at youtube.com/Southern IPM Center.